Hello everyone, welcome to part 2 of Tumor Immunology. In today's lecture we're going to be understanding the mechanisms actually by which tumors are going to be avoiding the immune recognition. Also we're going to be having a sneak peek on natural killer T cells, which are actually rather interesting. So let's get started. First of all, the mechanisms by which tumors avoid the immune recognition are plenty. There are multiple ways, and we're going to be talking about them shortly. First one is going to be by low immunogenicity. So first of all, we've got tumors are going to be having no peptides of novel proteins that can be present by the MHC to allow the immune system to detect the irregularities. So basically, if our T cells are not going to be detecting these abnormal peptides that are going to be presenting by the MHC class 1 naturally, then our immune system is not going to be able to know that this cell is a tumor cell or it is irregular in any sense. Therefore, there are no actions that are going to be taken against these cells, and therefore these tumor cells are going to go unnoticed. Next, also, there will be no adhesion molecules or co-stimulatory molecules. Now, this is very important, by the way. Even though this is present as a side point, I'd like to elaborate on it further. We've got no co-stimulatory molecules. In the absence of these co-stimulatory molecules, the cells in the first place are not going to get activated. So even though we have a signal, even if the MHC is going to be binding to the T cells, without the presence of the co-stimulatory molecules, giving some examples like the CD7.1 and the 7.1 and the 7.2, it's going to be, it's the B7.1 and the B7.2, then there is no reaction that is going to be taking place. So this is extremely important. So these factors are also important in the signaling of the cells and the activation naturally. Next, essentially the immune systems will not be able to identify these cancer cells. Therefore, they're going to be, just like we said before, go unnoticed. Next, the tumor is going to be treated as a self-antigen. Now, this is actually very interesting. First of all, we're going to be having, we have to recall that the TCR-CD3, which is the main receptor that is used by the T cells to recognize other cells, it's going to be binding to the MHC class 1 complex. Next, the co-stimulatory molecules are also going to be binding, just like we mentioned a couple of seconds ago, where we have the co-stimulatory molecules that are going to be making sure that our signal is getting across. So what happens here is that those MHC class 1 molecules will be lowered and the co-stimulatory molecules are going to be absent. So we can immediately actually understand that if the MHC class 1 molecules are not going to be present, or they're going to be lowered, then naturally our immune system or the immune reaction, by in this case the activation, is not going to happen. Also, we mentioned here that the co-stimulatory molecules are going to be absent. This also there's no way that we're going to be having an immune response unless we have these co-stimulatory molecules. So these are two major ways that are going to be blocking this, blocking the immune system from understanding, or it's going to be blocking the immune system from inducing an immune response against these cells. Also, the T cells are not going to be activated, and thereby the tumor cells can be considered as self. So we just mentioned that. Next, we've got the antigenic modulation. Internalization and degradation of the antigens that trigger the immune response are going to take place, rendering our immune system unable to identify the tumor cell. This is actually very interesting. So basically, we've got certain molecules that are present on the surface of the cells that our immune system are going to be detecting, and therefore classifying this as an irregular cell. So what's very interesting here is that these antigens that are going to be present on our cells, or the tumor cells, are going to be internalized. They're going to be entered into our cell and degraded. Therefore, our immune system will not be able to detect these irregularities. Therefore, no immune response is going to take place. That is actually my favorite, honestly. Next, we've got the tumor-induced immune suppression. Tumor cells are going to signal to Treg cells to attach to them and release TGF-beta and interleukin-10. So now let's understand this. By the way, these are both inhibitory cytokines. So what is going to be happening here is that we have a tumor cell. This tumor cell is going to be signaling to get these Treg cells. These Treg cells that are going to be attached to this tumor cell now are going to be releasing, just like we said, TGF-beta and interleukin-10. Since these are inhibitory cytokines, therefore our immune system is going to be suppressed to an extent. And also we have to note that not only do these Treg cells, they're going to be releasing these cytokines, also the tumor cells themselves are going to be releasing TGF-beta. Next, the tumor-induced privilege site. This is also very interesting, actually, in the sense that we have, for example, we've got a tumor cell at a particular location. It is going to be secreting certain molecules that are going to be protecting it. It's going to be hiding it. It's going to be masking it. Therefore, these cells are not going to be detectable by our immune system. Some of these factors that are actually produced are called include the collagen. Natural killer cells are the most important cells to combat tumor cells. Essentially, they're going to be checking out all of our cells in the for the presence of the MHC class 1. This is very important, by the way. Like these natural killer cells, they are going to be moving randomly between all the cells that are present around them, and they're going to be detecting the presence of the MHC class 1. In case it's present, then these cells are going to be normal. If not, then these cells are going to be attacked. How are they going to be attacked? These natural killer cells are going to be killing these cells by the induction of perforins, which are going to be naturally inducing pores into our cells, therefore killing them. I mean, well, due to the imbalances, therefore it's going to get swollen and pop. And we've got granzymes. Now, something to keep in mind here is that natural killer cells have two main receptors, the activating and the inhibitory receptor. The activating receptor is always going to be binding on its ligand, whereas the inhibitory receptor is going to be binding to the MHC class 1. This is why in the absence of MHC class 1 is very important. Now, I'd like to just address the image that is present on screen at the moment. We've got a target cell that is going to be having the MHC class 1, and then we've got going to have the activating ligand. This is incredibly important, by the way. So on the natural killer cell, we've got this inhibitory and activating receptor, just like we mentioned. What is interesting here is that we're going to be having this inhibitory receptor in case the MHC class 1 was present. This inhibitory receptor is going to be binding to this MHC class 1. Therefore, there's going to be inhibition, and these natural killer cells are not going to be killing these cells, these target cells. However, if these MHC class 1 were not present, then these inhibitory receptors are not going to be activated. Therefore, the activating receptors are the only ones present, therefore killing these cells. To understand the concept mentioned before on the molecular level, we have to know that upon activation of the activating signal, the protein kinase, now we're talking, by the way, at the molecular signal. So upon activation, there's going to be the protein tyrosine kinase is going to get phosphorylated. When something gets phosphorylated, for the most part, it's going to get activated. So now this protein, kinase is going to get phosphorylated. Now this is when we have a positive signal, an activating signal. Next, if there is an inhibitory signal, then what is going to be happening is that there's another protein called the protein tyrosine phosphatase, and well, we definitely know that the phosphatase is going to be removing phosphate groups. Therefore, they're going to be removing the phosphate that was earlier activating, or going to be leading to the activating of this pathway, and it is going to be stopping it. This is actually very fun. Th2 cells can support by producing interleukin 4, which indirectly blocks a neoangiogenesis. I want to talk about this actually in more details in the interferon gamma and the importance of it. First of all, it's going to be inhibiting angiogenesis, therefore the cancer cells will not be able to get nourished. Now, I want to elaborate on this a little bit further. Well, we know that angiogenesis is essentially where they're going to be forming new blood vessels. These blood vessels, since we know that the cancer cells or tumor cells, they're going to be rapidly replicating, they're going to be active in abnormal levels, therefore they typically need nourishment, and therefore they are going to be producing these blood vessels. Now, what is very interesting about this interferon gamma is it is going to be inhibiting this angiogenesis. Therefore, there are no blood vessels that are going to be formed to nourish these tumor cells. Therefore, if they do not get nourished, naturally they're going to be dying, or they're going to be suppressed. Next, increases the tumor cell MHC class 1, which will allow the cytotoxic T cells to identify them. Now, this is where it becomes a little bit confusing, because earlier we mentioned in the presence of the MHC class 1, the natural killer cells are going to be not activated. Therefore, now we are knowing that the MHC class 1 is going to be allowing the cytotoxic T cells, which are different than the natural killer cells, are going to be identifying with the presence of this MHC class 1. So it's important to know that there are two different pathways that one might use this to get inhibited, and one might use it to get activated. Next, it's also going to be, the interferon gamma is also going to be activating macrophages. Now, in the last part of our lecture today, we're going to be talking about the natural killer T cells. They play important roles in both the innate and the adaptive immune responses, as well as the anti-tumor responses. They are going to be co-expressing the alpha-beta TCR-CD3 receptor. By the way, this is very important because we know that the vast majority of the TCR-CD3 receptors that are primarily present, obviously, on the T cells, they're going to be having the alpha-beta. However, there's another type of TCR-CD3 receptors, which are the delta-gamma receptors. So basically, they're going to be having the TCR-CD3 receptor, which is a staple of the T cells. They're also going to be having the CD16 and the CD56, which are staple of the natural killer cells. Therefore, since these are both present, we call them the natural killer T cells. Natural killer T cells are going to be recognizing the CD1D, which is a receptor. I mean, this is going to be the ligand that is going to be binding to, instead of in the case of the normal natural killer cells, they're going to be binding to the MHC class 1. So you immediately can see this contrast. It is worth noting that they bind self and foreign lipids and glycolipids. Recall that in the MHC, we're only going to be presenting peptides. In this case, we're going to be looking at the lipids and the glycolipids. At the end, the major differences between the natural killer and the natural killer T cells, I want to stress that this is incredibly important, and there are some very important differences between them. First of all, the major ligand for the NK cells, or the natural killer cells, are the MHC class 1. I forgot to add it here, so it's the MHC class 1. While for the natural killer cells, they're going to be the CD1D. Next, the NK cells are stimulated by peptides. We mentioned this also earlier, where the natural killer T cells are stimulated by lipids. This concludes our mini-series on tumor immunology. Thank you for watching. This has been Ali from B Biology.